All right, so here we are moving on to a discussion of protein structure. Protein structure has a couple of different layers in almost like a hierarchy of how it is organized and described. So the first level is your, well, cleverly titled primary structure. The primary structure is just the amino acid sequence of a polypeptide, or the amino acid sequence of a protein. Now this is read from that N to C direction, and proteins with very similar primary sequences are generally going to be homologous and have similar, if not identical, functions. Now variations within those, also known as mutations, you've probably heard about like missense mutations and silent mutations. Well, that would be on your nucleotide level. You can have primary structural protein differences that can ultimately be lethal. They could be deleterious to the protein, or they could have no effect whatsoever. Now the next level of organization of protein structure is your secondary structure. Your secondary structure is composed of localized arrangements of that polypeptide. So we're talking about small portions of the protein and how they fold together to make unique structures. Now this is stabilized by hydrogen bonds within the polypeptide backbone. So different parts of that polypeptide backbone are going to hydrogen bond with one another. Your primary structure is stabilized only by peptide bonds, and those peptide bonds are your covalent bonds. So the most commonly observed types of secondary structural elements are the alpha helix and the beta sheet. An alpha helix, which we actually kind of looked at those a little while ago, those are those helical structures. They're known as alpha helices. They're rigid rod-like structures twisted into right-handed helical conformations. Now, the hydrogen bonds are going to be within the backbone that stabilize that, there are going to be hydrogen bonds between different parts of the backbone of these polypeptides. Beta sheets are composed of two or more polypeptide chain segments. So on the same polypeptide, but two segments within that polypeptide. When they line up side by side, you have hydrogen bonds between those backbones that basically make a beta sheet, which we actually also kind of saw sheets. Those are simply those arrows, and we're going to look at them again in just a moment, but those are your two major structures, alpha helices and beta sheets. There are others, but they're significantly less common. Okay, so here's some good representations of an alpha helix, which is on your left-hand side of the screen. And what I want you to see with that is, and this is kind of tough to tell, but the atoms that I want you to recognize are this blue one and this yellow one. So that would be kind of or you're going to see hydrogen bonds between these blue and yellow atoms. Here's one, here's one, here's one, here's one, and so on. And so it's these hydrogen bonds between different parts of the backbone that actually help establish that alpha helix. Now, the beta sheets are on this side of the slide. And beta sheets actually fall into two different categories. They are considered to be parallel or anti-parallel. Now, the difference between those is whenever you think about the direction of these polypeptide chains. So if you look, we've got on model A here, we've got N and C. But then the opposite strand or the opposite segment is going to be in N to C direction. Whereas a parallel strand has both N and C regions of a polypeptide basically lined up. Now, the difference between these is that your anti-parallel sheets, look at those. Those hydrogen bonds are straight. They're straight, and if you compare, let's go ahead and I'm going to highlight this hydrogen bond to looking at a parallel option, this hydrogen bond right here. That hydrogen bond within the anti-parallel representation, that would be, well, it's straight and it's shorter. Now, what do we know about bond strength whenever it's a longer bond versus a shorter bond? Well, those double bonds, for instance, are very short, and they are very strong. The shorter that the hydrogen bond is, the stronger that it is. So that ultimately gives way to the idea that anti-parallel beta sheets are stronger and more stable than parallel beta sheets. Now, your next level beyond that is going to be the tertiary structure. This is a unique three-dimensional conformation of a fully folded compact polypeptide chain within a protein's native structure. Okay, so essentially what this is you could kind of think about it as we've been building up. So we have covalent bonds of our primary structure, covalent bonds that establish our primary structure. We have hydrogen bonds of the polypeptide backbone that establish the secondary structure. Tertiary structure is going to be an aggregation of those two things. So tertiary structure is in part stabilized by covalent and hydrogen bonds, but ultimately the native structure is going to be, well, basically dictated by lots of hydrophobic interactions. But before we get to that, protein folding is a process in which an unorganized molecule acquires a very highly organized structure. So if you think about like a globular protein, and everyone always likes to take like, here, this is from a fruit by the foot that I had a little while ago. If you haven't a fruit by the feet, well, they're still pretty good. They were good when I was in 3rd grade, they're still good now. So let's imagine that this is this would be a polypeptide, an unfolded polypeptide. Well, covalent bonds keep it intact. Then we got hydrogen bonds that kind of establish some regions that are folded, but then whenever I personally think about a tertiary structure, oh, there's my tertiary structure. Okay, it's folded. And this might seem like, oh, well, that's just chaotic, and oh, it's clumped together. But it's a very organized structure. Now, how is that dictated? Well, it's dictated by interactions between side chains, or R groups, of these amino acids. Now, residues that were very far away from one another may now be in very close proximity. So if we look at, here, this end right here is my N termini, this end right here is my C termini. Whenever it's folded up into its globular structure, now all of a sudden a residue on my N termini is right next to a residue on my C termini. How about that? They're right next to each other. Now, most water molecules are going to be excluded from the interior of the protein, and that's just a consequence of presence of polar groups versus nonpolar groups. It's going to be lots of nonpolar groups at the center of a protein. So large globular proteins are going to basically be broken down and organized into things that are known as domains. So interactions that are going to stabilize your tertiary structure. Hydrophobic interactions. These are going to be the driving force behind protein folding. They're going to exclude water from that interior of a protein. Electrostatic interactions or salt bridges, which if we remember salt bridges, those are salt bridges are electrostatic interactions and hydrogen bonding. There are also going to be instances where there are simply hydrogen bonds that are established. Now, protein folding is a thermodynamically favored process. So the delta G is going to be negative for a protein going from its unfolded state to a folded state. Covalent bonds are also going to stabilize tertiary structure. However, it's not extremely common because in terms of covalent bonds, well, we've got disulfide bridges. Disulfide bridges are between two cysteine residues. So we don't have a huge number of amino acids that are going to be involved in formation of any sort of covalent bonds. And ultimately, these are going to help stabilize proteins against changes in pH and salt concentration. Okay, so cysteine has that sulfhydryl group that's very reactive. And one of the more common reactions is a reversible oxidation that forms a disulfide bond. Okay, so what we've got here at the bottom of this is we've got our there's one cysteine residue, here's another cysteine residue. Those two residues, well, they've got this nice little sulfhydryl group. And what ultimately happens is they both get oxidized and form this disulfide bond, or this disulfide bridge. This is something that's a very strong bond. It could care less about the pH of a solution, and it's going to help stabilize polypeptides and proteins. Yes, there we go. The fourth and highest level of protein structure is known as your quaternary structure. Now, the tricky thing about quaternary structure is not every single protein is going to have the quaternary structure, and the reason for that is your quaternary structure consists of, your quaternary structure consists of multiple polypeptides folding together. So not every protein is going to have a quaternary structure because not every protein is going to fold as a part of a complex with other proteins. So this is found in many proteins, but not all of them, because of this requirement right here. You've got to have more than one polypeptide chain. Now, the types of interactions between the subunits is the same as the tertiary structure. So you're dealing with hydrophobic interactions, salt bridge formation, hydrogen bond interactions, a whole host of other interactions. Your summary of your different levels is displayed right here. So here would be your primary structure. Gives you this nice randomly folded piece of string. Secondary structure, well, then you might see like a helix or a beta sheet, or a pleated sheet is what they're commonly known as for. Tertiary protein structure, well, that's when that protein folds as a whole, and it looks like, yeah, sure, you can pick out the alpha helices or maybe a beta sheet, but there's more to it than that because two proteins fold together to form one thing. All right, well, I'll see y'all in the next video. Have a good one.